From Microbe TV, this is Office Hours for Wednesday, the 23rd of April, 2025. I'm Vincent Racaniello, and welcome to your viral corner of the internet, where we give you science, information, and truths, the opposite of what this administration peddles on a daily basis, shamelessly, I might add. Welcome, everyone. In particular, welcome to our moderators. Tonight we have Les, we have Vanity Nutrition, we have Steph, and we have Tom Steinberg. Thanks all for coming. Tell us where you're from. I'll shout you out. Once again, last week I forgot to show you Dixon's photographs. It just get carried away in, the nonsense that's going on in this country. It's just incredible nonsense. I heard the president of Harvard saying that, it's one thing if you want universities to avoid anti-Semitism, we can work on that. But then to want to control admissions and faculty hiring and subject and all that, what does that have to do with anti-Semitism? It has nothing to do with it. It's completely unconstitutional to require that. And this administration thinks they can get away with anything. Now, unfortunately, it's going to take a long time to get all this back. But I'm confident that you can't just walk to a university and say, I'm going to take all your money away unless you do as I say. I mean, yeah, there are a couple of Supreme Court justices who would agree with that, but sanity says it's not true. Dagfin is in Oslo, 2 a.m. Welcome. Tom is in Western Oregon. We had a nice TWEVO today where we talked about the dire wolf story. It was not de-extincted. They took a couple of single nucleotide polymorphisms in the dire wolf genome, in genes that they thought may make dire wolf qualities, put them in the gray wolf genome, and then implanted that DNA in a nucleus of a gray wolf, and then into a surrogate dog it went. And now we have a couple of animals who are basically gray wolves with some single nucleotide polymorphisms from dire wolves. They're not dire wolves, folks. Whatever you want to think, they're not de-extincted. And probably we shouldn't de-extinct animals because they went extinct for a reason, and the environment now is totally different. Anyway, go listen to that TWEVO. It was very good. MN is in D.C., where it was a nice day. Will is in China, 21C and raining. Kip, thank you for your contribution to science communication. Kip writes, malignant incompetence, when a mistake is made which has major downstream adverse effects, HHS under RFK. I would say in every aspect of this administration, there's malignant incompetence. Just picking tariff numbers out of a stupid formula is malignant incompetence. The economy is being destroyed, folks. You know that. Anyway, the phrase malignant incompetence is wonderful. As David Gorski wrote that over at Science-Based Medicine. And it's exactly what's going on. These people are incompetent. None of the heads of whatever cabinet members are competent for their position. They all do malignant incompetence. And by the way, speaking of attacks on universities, every one of them went to some Ivy League university or similar. And now they want to destroy them. Good for you. Where are you going to send your kids? Oh, Vanity had a quote here. It didn't show up in the chat, but Vanity had said, some of the anti-vax propagandists who have ties to RFK Jr. are saying a bioweapon started the measles outbreak. This is so screwed up. You don't know anything if you think that. It's disgusting to hear and appalling that his position gives him the platform. Yeah. You can't, no, it's not a bioweapon. It's freaking measles virus, for God's sakes. It's causing measles. Why would you need a bioweapon? It doesn't make any sense. These people were unvaccinated, dumbass. Oh my gosh. Anyway, so the reason I pulled that up, John writes, whenever I hear people talking about bioweapons, I know they have a background based on fictional movies and not science. Right. They don't know anything about science whatsoever. Will says, a federal judge just accused Department of Justice lawyers of falsehood in court. If lawyers lie, you can bet most coming out of the White House and lackeys are lie. Well, lawyers have lied to some extent always, right? And not every lawyer, but many. And anything that, if you listen to the press secretary, she lies left and right. It's just disgusting. It's just people just, what are you going to do? Yeah. Did we say Steph was here? Well, if we did, it doesn't matter. Twice Steph. Welcome, moderator Steph. Visto is in Sydney, 20C. Oh, and did you see this? The White House changed COVID.gov to their page saying SARS was from a lab leak, with no data to prove that. Zero. And no scientists involved. Zero data to prove a lab leak of any kind. Just rumors and innuendo. You know what you call an Italian suppository? Innuendo. It's all innuendo. Hello, Ju in Austin, Texas. 77F is sunny. Kang is in Chicago, 19 and clear. See, it's getting moderate now all around the Midwest and the East. John is in Minneapolis, having a fire because it's 18C. Brian is in Montreal, 10 degrees and sunny. Carol, our favorite nephrologist, Bakersfield, California, 8127C. Tom, tomorrow will be 75, and so will your twin brother. Well, happy birthday, Tom. You don't look 75. Good for you. I just came across pictures of us at ASV that time in Wisconsin. That was a good time. Leo is in the Mendocino Coast, 12C. Noir is in New York City. You're actually in New York City. Wow. Good for you. You're going to come visit. Jan T is in Tucson, 31C. Mike and Lori are in Seattle, 7C, so a little chilly there. Costella is here, but I'm out. 1000X love. Thank you, Costella. This is how you say happy birthday in Chinese. Cool. Zhu ni sheng ri kuai le. Sorry to butcher it. Cheryl is in Southern Ontario, 19C. Mismatched week while getting ready to drive to Tours in France to take our 97-year-old to see the chateau. That's cool. Chateau are beautiful. Elizabeth is in West Virginia. She says, we will know the cause of autism by October. This is just laughable. It's not how science works. You don't set a timeline to have an answer to a scientific question. I'm so mad. Yeah, it's horrible, Ju. They just don't know what they're talking about. And as Paul Offit said, he's going to make something up about vaccines and autism and say, I got the solution. It was being hidden all the time by the FDA and the CDC. How do you fight that? Saying they had these data all along and I found them and vaccines cause autism. This is ridiculous. He will use that as an excuse to cancel vaccines. Cheryl is in Santa Rosa, 62F. Rainy, south of Raleigh, 70F. Pete is in London, 10C. And Mike, it was 17C, not what he said. Margot is in Phoenix Metro, 89F. Peter says, speaking of Harvard, I'm here right now. Was at 2 talks today. Good for you. I'm glad the talks go on. Jonathan is in Encinitas, 16C. Chris and Lisa in Montreal area, 20C today. That's pretty mild. Jonathan says, malignancies destroy the host system due to unregulated extraction of resources. Yes. We see your posts, Pete. No worries. Cheryl listens to Dire Wolf. Every time I hear Dire Wolf, I want to hear Dire Straits. That's all. Yeah, it was fun. SRR is in Paso Robles. Thank you, SRR, for your contribution to science communication. Environmental, I don't know. I think we're in a lot of trouble. Yes, we are. And, you know, it's 2 years to midterms, and I think we could flip Congress then, but that's too long. Everything will be broken by then. It's just horrible, yeah. This is what you did, folks. This is what you and your vote did. Environmental. On Earth Day, the idiot president said they had a president that believed in science. This is just a lie, another lie. Nothing coming out of his mouth can be believed, except when he says his name. Patricia is in D.C. Chris, thank you for your contribution to science communication. Really appreciate it. Thomas is here for 30 seconds for a good laugh. This is not disappointing so far. Good for you. I'm glad. Yes, good for you. EHS Talent is in Lehigh Valley. Elizabeth says lawyers lying to the public is a totally different thing from lying in court. There need to be consequences for these people. The problem is the DOJ and the FBI are all loyal to the madman. What can you do? I mean, they wouldn't prosecute Signalgate, which is a prosecutable offense. And he did it again, a second time. Because he's an idiot. He doesn't get what security is. And now there's indications that a lot of intel is leaking out of the U.S., probably to Russia and China, because they have destroyed the security of the computer systems, with Musk screwing around with it. Patricia says, many are saying COVID is now like the cold. What do I tell them? Well, it is not yet like the cold. It might be in 10 years. But, you know, 800 to 1,000 people a week die in the U.S. Is that a cold? That many people do not die of colds. No. We still have a lot of vulnerable elderly population. And don't forget, younger people can get long COVID. So it's not like the cold. Where did they get that crap from? Oh, my cousin got COVID and it was mild. So they make a broad conclusion that COVID is now like the cold. That's not how it works, folks. Your experience is meaningless. I'm sorry. Many of you do the same thing. Oh, I had COVID. It was mild. It must be mild now. No. Just listen to Daniel Griffin moan about the deaths every week. Why aren't you listening to Daniel Griffin? It's 30 to 40 minutes. Learn something. Patricia, at what point is a novel virus no longer novel? When it's been in people for a while. I don't know. So you want to know if it's going to stay in people, so you give it some time. A year. It was novel when it first went to people because we'd never seen it before. But then in a year it had been through around the world. No longer novel. We know about it. And now, of course, it's not novel anymore. Coronavirus infectious disease. Yeah. COVID. it's not I wouldn't call it a novel coronavirus any more than you would call SARS-1 a novel coronavirus, although back when it came out, we did. Tim is in Toronto, 10C. Environmental says, I'm using a nasal spray called Profi that was suggested to me as somewhat barrier to some aerosols. Do you think they work? No. If it's not been through a proper clinical trial, it doesn't work. Oh, Noir's in the ER. I hope you're okay. Please get better. Drinking grape-flavored vancomycin. Hope it's okay. Vanc is a tough one. It's hard-hitting. Serge is in Ottawa, Canada. Joel is in Bath, UK. I've been to Bath. It's a cool place. I had a good day. Hank says RFK Jr.'s autism registry is reminiscent of how the Nazis' T4 euthanasia program got started. You know, we talked about how RFK Jr. last week said autism destroys lives. People with autism can't have normal lives. They can't have a job. They can't do anything. And a lot of people were pissed off about that. And I just want to say, I have a son with autism, and he's working. He has a job. He's fine. He's got issues, of course, but his life isn't destroyed by it. So I resent that, RFK Jr. You don't know what the hell you're talking about. Now, I know my experience is irrelevant, but a lot of people with children with autism spoke out and said, no, collectively, our kids are having good lives. So don't say that because you don't know anything and you haven't bothered to read anything or study. He never reads a bloody thing. Go away, RFK Jr. Noir says, send bacteriophage, please. Oh, yes. That would be nice if we could do that, wouldn't it? And yeah, every week, Implosive, there is stuff to be upset with. It just gets worse and worse. Yeah. Oh, Harvard, you're not going to agree with us? I'm going to take away your tax-exempt status. What kind of a child is that doing that? This is the president of the frickin' U.S. He's being a baby. We're going to take away your tax-exempt status because you won't listen to us? Screw you. You can't do that. They don't have to listen to you because your demands are ridiculous. Carol, over the years, my patients have lost their renal allografts to BK virus nephropathy. Maybe a mini lecture on BK. Yeah, that would be interesting. Everybody has BK virus, yes, and then when you get immunosuppressed, it replicates. Yeah, very interesting. Yeah, we can do that. BK virus. Why would he want a registry of autistic people in the U.S.? Is that a precursor to gathering more medical info about Americans? I don't think you need a registry to do a clinical trial. He doesn't know what he's talking about. He doesn't know how to do a clinical trial. He's never done one. He's never read about one. He doesn't even bother to study and read up what's in a clinical trial. Now, this one would actually not be a clinical trial. It would be an observational study, a real-world study, because we don't have any drug. We have people with or without autism. We'd like to know what's different between the two groups. So we have to find out. It's hard because there can be a lot of confounding variables because these populations have grown in different environments and they are biologically very different. Abdur Rahman is in Manhattan. Nice to see you again. Tom Steinberg says, Dire Wolf is a fine song from Working Man's Dead. Oh, I didn't know that. Cool. Elizabeth says, these idiot passwords were on the dark web within hours of Signalgate. It's all insane. But people still support these idiots. Yeah, it's all insane. They don't know what they're doing. They're totally malignantly incompetent. Visto says, is Beyond the Noise available as audio? No, I'm sorry, it is not. but you could play it on YouTube on your phone in your earplugs, right? It's the same thing. I didn't take any COVID vaccines. Should I start taking them now? Did you take any? Have you got COVID? Bad Cat is in Winthrop, Maine. Evans Forever at Campbell, this idiot who, right? We used to talk about Campbell all the time on Q&A with A and V. He's just says dumb things, and now he's with the lab leak. He doesn't know what he's talking about. You know, all you have to do is read the papers which have the evidence for a Huanon market spillover from animals into people. There's great evidence there, and there's no evidence for a lab leak. How would you have a lab leak? So a lab would have to have SARS-CoV-2. Almost this, that would say the same exact virus as SARS-CoV-2. They didn't have it in Wuhan. It's quite clear. They sequenced all of their isolates. They didn't have it. And then you'll say, they were lying. Well, that's not science, folks. Don't give me that crap. It's not science. They didn't have it. End of story. It did not leak out of their lab. Oh, maybe they didn't know they had it. They sequenced everything in their freezer. Do you understand what that means? Stop bringing up these straw horses, or whatever you might call them. RFK will be violating HIPAA. How would he be doing that? Environmental, explain. Aubrey, hello. In Richmond, it is about 68. Seems dumbassery is running rampant throughout the U.S. Yes. We are dumb. We are not dumbassery here at Microbe TV. We see science because we are all scientists. And no, we are not brainwashed. We are not under the pay of Big Pharma. We are scientists trained by the scientific method. And we know what's BS. And everything they are promulgating is BS. I don't get money from anyone. So you can't blame me that on my behavior. Pete says most of us here are TWIV listeners. Problem is, how do we convince others? I don't know. Make it personal. Have a conversation with them. Why do you think it came from a lab? Oh, well, this government study said so. Well, then you could say, did you read the government study? No, I just read the conclusion. Well, do you know what the evidence is for a lab leak? No. Well, they were doing gain-of-function research. Do you know what that is? No, so you can explain it to them. And they were working on viruses distantly related to SARS-CoV-2, so it couldn't have leaked from the lab. And they'll say, oh, I don't know, the FBI, CIA, blah, blah, blah. It's hard, especially with this stupid web page with a picture of Trump and between the words lab leak. Why do they have to put his picture there for? What does he have to do with it? He knows nothing. He knows less than nothing about lab leak. Patricia says, if you know, is HPV not recommended in men and women under 45 if they've had many partners? I don't know. I'm not aware of that. I could ask Daniel. You could send an email to daniel@microbe.tv, and I will put it on the list for tomorrow, Patricia, because you're always here, and we'll answer it, and you'll hear it answered on Saturday. Lily Siso Amuna, I have the same experience. Autistic people often have the most wonderful and beautifully stunning minds. Did you ever see the TV show Astrid? Right, it's about an autistic lady who works with the police to solve crimes. And it is a TV show. It's fiction, right? But, I mean, she has special talents that neurotypical people don't have. Sam likes the content and enjoys consuming the videos and knowledge. Thank you. I'm glad you do. Mario is in East LA, 17C. Thank you for the great SciComm. More prominent people in science should be outraged and start broadcasting it more loudly. Yes. Yes, I think many people should be doing, scientists should do this, but they're not. Hello, Martha from Charleston, South Carolina, 21C. Yes, this is a good point, Elizabeth. Autism registry being pushed by people who are viciously against any gun registry. The hypocrisy. Total hypocrisy. Kang wants to know, what is the worst, Great Barrington Declaration or the lab leak? Well, I don't remember the whole declaration. I just remember, in my opinion, one of the most offensive parts was to let it rip. We don't need to be masking. We don't need to be shutting down society. Just let it rip without any knowledge of the virus and what it could do. These are not scientists who are saying that. Okay, Jay Bhattacharya is not, he's a health economist. Okay, so if you think an economist is a scientist, it's fine, but he doesn't know any biology. So he said, let it rip, and so did all the other signers. And that's a bad idea because we didn't know about long COVID then, right? Stupid, really stupid. And you have to exercise caution when you have a new agent that you don't know anything about. And these people, they wanted society open because of economics. He's a health economist. He wanted the money to flow again. It's so smarmy. Visto, I see typhoid bacteria have become extremely drug resistant. Could phage help? Yes. People have to work on it and find phage that could help. But it's tough for because he's like typhoid. Your average lab leaker will be stumped when you ask for a definition of phylogeny. Yeah, well, they wouldn't even ask. You say, okay, if you look at the phylogeny of SARS-CoV-2 and RITG13 and SARS-CoV-1, they're not very related to SARS-CoV-2. And they wouldn't even say, what's a phylogeny? Because they have no curiosity. They just want their viewpoint to be right. And Bad Cat says, in Central Maine, friends are getting docs after finding ticks. I had Lyme three times. First time was terrible. Why can dogs get Lyme vaccines but not people? The Lyme vaccine was taken off the market. Had some problems, right? But apparently the dog vaccine is okay. House of Tonic Live says, what was Jay Bhattacharya referring to about dropping life expectancy? I don't know. Let's look it up. Jay Bhattacharya dropping life expectancy. I get an article. Bhattacharya was dangerous. Now he's leading the NIH. So, there's been a decline in life expectancy in the U.S. In the past years. And so it declined. This was in 2019. It declined a tenth of a year to 78.6 years. Well, of course, COVID pushed it a couple of years down further. And so he has weighed in on why it is declining. And I don't care what he felt. I have no interest in what he thinks. Jason, hello from France. Is HPV any good after exposure? It It depends on how far the infection has gone. It is still efficacious at preventing cancer, but it's recommended that you get it before sexual activity because that way you won't already have been infected. But it still can be taken and still has some efficacy after exposure. Power of podcasts cannot be understated at this point in time. Yeah, there are a lot of political podcasts that are very good, right? I don't watch, but I know people who do. I don't remember the names. Sorry. The lovely town of Great Barrington has to be smeared with the nonsense declaration. Yes. You should sue to have the name removed from it. Hello, Patricia from Massachusetts. Hello, Julie from North Central Pennsylvania. Maureen is in Northeast Ohio. 70F. Riptocomp is in Boulder. Will has renamed Bhattacharya to J. Let it rip in the wider community to enable non-existent herd immunity to protect the vulnerable. Yeah, he said herd immunity was going to be the solution, and so did Marty Makara, who also doesn't know anything. Bhattacharya is a conspiracy theorist, Thomas says. Okay. That's the end of the questions so far. There are going to be many more. But I want to take this moment to show you pictures of some of Dixon's watercolors, okay? I've been promising to do this for a while. Okay, so I have a bunch of these watercolors from Dixon. He gave these to me. He had put on an exhibition at Fordham University in New York City, and after the exhibition, he gave them all to me, and about 20 watercolors. So I put these three up over the fireplace at home. He drew a lot of these plant-like entities. They kind of look to me like cattails. He drew them in different shapes. So I thought the three of these went well together. They're all brown, obviously, but different shapes. So those are three of his works. And then he also did different things, right? This is a watercolor also, which is very simple, but I like it. And then back to the plant-like things, the cattails, if you will. This one is in black and white, so that's quite interesting. And I think I have one more. This one is another version in monochrome of these cattails. I call them cattails. He called them plants. Other people called them. Some people said they were fungi. So there you go. I do have some pictures of his other works. I have to find those because I do think he was a pretty good artist. Nevitz says the HPV vaccine is against 9 serotypes. Right. So if you got infected by one, you could still benefit from vaccination. Absolutely. That's the key. Thank you very much, Nevitz. Crowdsource the answer. And John says the CDC website still says to get the vaccine if you've never received one. You should get the full course of 3, I guess, right? Reduction of life expectancy was due in part to fentanyl. Also due to SARS-CoV-2. I'll show you. I showed this slide in the vaccines lecture. So let's pull up the vaccines lecture. We'll do a vaccines lecture here as a mini lecture at some point. We're working our way slowly through. Don't worry. We have plenty of time. What was vaccines? Was it transformation? It's 18. Come on. Vaccines. Okay. I'm pretty sure I had a life. Yeah, there we go. So as you all know, let me share this. As you all know, life expectancy since 1900 has increased in both men and women until in the early 2000s. For women, it was almost 80, and for men, about 75. But from 1980 to 2020, it dropped at the onset of the pandemic. There are little blips over the years, some of which could be fentanyl. But look at that. It's a 1.5 year drop in life expectancy in 2020. And I don't know what's happened since then. I haven't looked for other studies, but here's the reference for this one. What is interesting is the difference in life expectancy by ethnicity, right? Look, black, white, total population, Latino. They have a longer life expectancy. That's very interesting. I don't know why that would be. Good. And this is the health inequity, less health life expectancy, which is unacceptable. And I don't see it getting better anytime soon. All right. Let's go back to me. So that's life expectancy. I'm lucky I can remember where I have all these slides. Okay. Polka Dot wants to know what I think about the new COVID MERS virus. Well, those are two different viruses, so could you clarify what you want to do? Patricia wants to know, are there any further updates? Variants continue to emerge on a regular basis. CDC in the U.S. tries to keep up with a variant-specific vaccine on a yearly basis. If you follow the CDC recommendations, you will get that. Most people are not wearing a mask. I still do on public transportation because it's very crowded. Tim says, I got the Prevnar 20 vaccine today. While talking about vaccines with my lovely nurse practitioner, she commented on how many vaccine skeptical families are asking for measles vaccine. There is hope. No, I think we're going to have many more cases and many more deaths before we see a real sea change in these people's attitudes. I just don't get it. Privyet, Veronika in Moscow. Welcome. Good to have you. Mario wants to know about the new chikungunya vaccine. We should do a TWIV on it. I don't know what's been published, but I'll look it up, and we will do a TWIV on it. That's a good question. Patricia, the Latino paradox, highest burden of diabetes, longest life expectancy. Red Hen 689 tells us that the Valor Lyme disease vaccine trial is supposed to report results at the end of 2025. Very good. 3-dose series with 6,000 people. We'll see. Good. Yes. You notice that FDA fired all the people involved in checking milk. So milk's not going to be checked anymore. I guess the states will do it. But, oh my gosh. It's just malignant incompetence. Yeah, there you go. It's ridiculous. Why shouldn't they check the milk supply? You could let every state do it on their own now. Seems to be a 50-fold duplication of effort. WHO said the new COVID MERS had I don't understand that. I'm sorry. So Nevitz continues to mask and MN. So I'm wondering, do you think in the summer months it would, because you still get, I guess you could still get infected because it's really hot in that mask. Today in particular, it gets hot. I have trouble breathing. Spartavus, I'm okay. Thank you. Nice busy day today. 2 live streams in a class. I love it. Jason said, a new neighbor here told me his wife got terrible kidney infection after COVID vaccine. She lost her kidneys on dialysis now. I don't know the details. So we have to be careful with these anecdotes. Why don't we ask our nephrologist what she knows about kidney failure after COVID vaccine. I've not heard that, but our favorite nephrologist, Carol. No, forgot your name. I'm sorry. I'm scrolling through. I can't search. Maybe she left. Carol. Yeah, it is Carol. Do you know anything about kidney failure after COVID vaccination? Thank you. Kang wants to know what part of the red blood cells do flu viruses bind to. Sialic acids that are present on proteins, of course. Doesn't matter what the protein is. And it's alpha 2-3 or alpha 2-6, depending on. So if you got chicken red blood cells, they're going to have alpha 2-3. And if you get other mammalian red blood cells, they're going to have alpha 2-6. But it's just the sialic acid. The protein doesn't seem to matter much. And Kang follows up, our patients with certain blood groups protected. No, they're not. There's no hemagglutination there's no viremic phase of flu. Okay, so there's no hemagglutination. That would be a problem. You get aggregates of red blood cells. That could be a problem. Then the ACIP just scaled back their recommendations for COVID vaccines. I don't recall that from Daniel's last report. Maybe I missed it. I'm getting all the same questions again. Why? Repeated. Zhu, thank you for your contribution to science communication. Environmental. Do you have any idea if Novavax will be approved? No. It sounds to me like it's not. Because they said they needed more data. And Novavax says we have all the data we need. So kind of an impasse, right? I don't know what's going to happen, and neither does Daniel. Yeah, I know masking isn't always comfortable, but it's worth it not to get sick. Shu, any plan on doing another live TWIV? I was there at the 1000th episode and loved it. Yes, I recall. We have, We're going to be at ASV in Montreal. That's the next live event, but nothing after that is planned so far. No. A Mpox was found with a furin cleavage site. Okay, well, I'm not surprised. So Mpox would be the MERS-like beta coronaviruses. And so these are there. You just have to find them. And the idea that the furin cleavage site was a signature for human manipulation of SARS-CoV-2 is ridiculous. Ridiculous. Carol says, I've never seen kidney failure from the COVID vaccine, but I've seen kidney failure from COVID infection. My daughter-in-law's mom is on dialysis after having COVID infection. Okay. That would make sense. Thank you very much. The difference between alpha 2, 6, and 3 is merely nucleic acid positional isomers, right? How it's connected to the next sugar in the chain. It's very subtle, and it makes a big difference. It's amazing, isn't it? Yeah. Love it. How's Amy doing? I can't say, right? Our conversations are confidential. You can imagine. You can imagine. Okay. At that point, that's a nice break point. Let's do a mini lecture. This one's going to be mechanisms of pathogenesis. It's got to be part 1 because this is a long topic and I didn't want to cut anything out like I usually do. Because I think you can understand it. It's pretty straightforward stuff. Not a lot of molecules and formulas. So what is pathogenesis? It's all the processes that lead to disease. A pathogenic virus causes disease, and a non-pathogenic virus does not. Pathogenesis is often a collateral outcome of the parasitic nature of viruses. What does that mean? Viruses are parasites. They take from the host, right? So that causes damage. That causes disease to a certain extent. But I want you to understand that pathogenesis is not an evolutionarily selected property. In other words, evolution doesn't select for more pathogenic viruses. The only thing that's selected for is fitness, the ability to reproduce. I mean, many things can control fitness, but not pathogenesis, not the ability to cause disease. And many people do not understand that. Oh, the virus evolved to become more pathogenic. No, you don't have any evidence for that. Maybe it became more fit, which means higher replication, and that may help transmission. So that's a nice selectable feature. And making more virus can cause more disease. So it's a collateral. It's not directly selected for. Okay, to study pathogenesis in the lab, we need animal models because in general, we can't infect people. There are some challenge experiments you can do. And you can make some observational studies in people, like you can measure virus at different times after infection and so forth, where it is. But, you're limited into what you can sample. You can't, blood is very easy. The upper tract is easy. Then anything else is tough. A lymph node biopsy, it's tough. You can collect feces, but it's not telling you the whole story. So we rely on animal models. The problem is mice lie and monkeys exaggerate. What do I mean by that? But the animals are just models. They're not predictors of what's going to happen in people. When we do preclinical studies on, say, an antiviral, we test them in an animal model. Sometimes it works. If it doesn't work, goodbye. But if it works, you still have to test it in people. Why? Because animals do not predict what's going to happen in people. If they did, we wouldn't have to do clinical trials. Well, we'd probably do them anyway. So there are all kinds of animal models. Here in the middle is a mouse, but there are all kinds of other animals as well. Some human viruses will infect mice or other animals, and that's great. We can study them. But the original SARS-CoV-2 did not. It had to be modified. But sometimes we can use animal models that model human infection. So there's a gamma herpes virus 68 of mice that's used to model herpes simplex virus infection. It does very well in mice. And there are other ways we can, instead of directly infecting, in addition to directly infecting, we can do modifications of mice. For example, we can make transgenic mice synthesizing human receptors, and that sometimes makes the animals infectable with your virus. You can give them human tissue transplants. You can modulate the host response. You can delete immune cells or antibodies, or you can overproduce them. So there are many ways that you can modify mice in particular, because they're genetically manipulable and they're small, they breed quickly, so they're good models. But not everything does well. Flu viruses don't do well in mice. So you need to use ferrets. Bigger, more expensive, harder to work with. And they're not people. So the actual full statement here is mice lie, monkeys exaggerate, and ferrets are not people. That's the saying. Now, speaking about transgenic mice, I want to push the work done in my lab in the 90s to establish the first transgenic animal model for a virus disease, the first transgenic mouse model for a virus disease. And so we wanted to make a small animal model for poliovirus, because the only animals you could use would be non-human primates, and I had no interest in working on them. So one of my first students, Kathy Mendelson, identified the cell receptor for poliovirus, right? It's a plasma membrane protein that binds the virus. So we had the gene for this virus. Sorry, we had the gene for this receptor. We put that gene into the germline of mice. We made transgenic mice. So the gene is in every tissue, and it's transmitted to the offspring of the mice. Those mice, now when you infect them with poliovirus, they get paralysis, as you can see here. Before you put the transgene in, the poliovirus receptor, if you infect them with polio, nothing happens. They're completely resistant to infection because they have no receptor. But all you need is a receptor, and everything else works. That's amazing, right? The receptor confers susceptibility to infection, but permissivity, that means everything beyond receptor that the virus requires, it's already there in the mouse. Isn't that amazing? This blew me away when this worked. Anyway, there are not too many other transgenic mouse models for virus disease. It just doesn't work for many viruses. So you can do it with SARS-CoV-2. You just put human ACE2 in, but you actually don't have to do that, because you can change 2 amino acids in SARS-CoV-2 spike, and that will reproduce in mice. Yeah. Transgenic mice, not transgender. Do your homework. You know what I'm talking about. A big part of pathogenesis is understanding what we call tissue tropism, the spectrum of tissues infected by a virus. Like we say, a virus that infects your intestines or the enteric tract is called enterotropic. A virus that infects the nervous system is neurotropic. A virus that infects the liver is hepatotropic. You get it? That's tissue tropism. Some viruses are quite limited. They only infect, say, the liver, but others infect every organ. We call them pantropic. So what controls tropism? Aha. Many things. First of all, susceptibility, which means the presence of the receptor. Here's some receptors for viruses, and if they are present on a tissue, the virus will bind to them, and that's required. No receptor, no binding and no infection. Permissivity of the cells is also important. Remember, everything past receptor binding is permissivity. Now you may say, well, Vincent, what are you talking about? Past receptor binding. Okay, the RNA gets into the cell. That's past receptor binding. Then, all right, let's call it a plus strand RNA virus. So it has to be translated into protein. So the ribosomes are required, the whole protein synthesis apparatus. And then you need energy, right? You need membranes, you need all of that. We've talked about that in a mini lecture before. So all of that is permissivity. Some cells just do not support the replication of a particular virus. Accessibility, cells have to be accessible. If you get a respiratory infection and the accessible cells are in your gut, the virus may never see it. Also, immune defenses are important. Sometimes immune defenses will just clear any virus, or in some tissues they're absent, and therefore you can get an infection established. For example, let's go back. Can I go back? So in these transgenic mice, the receptor, poliovirus receptor, is produced in many different organs, virtually everywhere. Yet, when you infect these mice, the only place poliovirus reproduces is in the central nervous system, in neurons. Why is that? Well, if you knock out the innate immune system, the interferon system of these mice, the virus reproduces everywhere. So interferon is regulating the tissue tropism of poliovirus. It's narrowing it to only the neural cells. And if you take away interferon, in particular type 1 interferon, now the virus can reproduce everywhere in the mouse. It's amazing. It's amazing. Now, influenza virus gives a great example of glycoprotein cleavage or spike cleavage as a tropism determinant, okay? The virus has spikes in its membrane, right? Just like SARS-CoV-2 has spikes, so does influenza virus. And this is a diagram of the spike, the hemagglutinin, in this red line here. It has to be cleaved. You know what cleavage means, right? No, not that kind. It has to be cut, okay? It has to be cut by an enzyme called a protease. And that cutting happens right here where this orange arrow is. And it exposes a hydrophobic peptide, which allows fusion of the virus to occur with cell membranes. So it's totally required for infectivity, that cleavage. Now, in human influenza viruses, the cleavage occurs after the viruses are released from respiratory epithelial cells. So an enzyme called tryptase cleaves the HA, so now it can go on and infect other cells within the respiratory tract. Now, that tryptase is restricted to the respiratory tract. That protease is restricted. So if some wayward influenza viruses got to your liver, say, they wouldn't replicate because there's no protease to cleave the HA. Okay. Now let's talk about H5N1 influenza. H5N1, the HA, the cleavage site is a furin cleavage site. Yeah, it's a furin cleavage site. Nobody put it there. It's always been there. Why is that important? The cleavage site in human influenza viruses is cleaved only by enzymes in the respiratory tract. That limits the tropism. But for H5N1 viruses, these furins are everywhere in your body. So this is why in humans, the H5N1 viruses can often cause systemic infection. They can infect many organs, okay? And that gives it a broad tropism. So just a change of a few amino acids at the cleavage site gives it a broad tropism. And you know, when this was first discovered in H5 influenza viruses in the 1990s, people didn't say it was introduced because that would be a stupid illogical thing to say based on no science. And that's where we are today with COVID. Stupid, illogical statements. And, my colleague virologists, if you want to make those statements, I will call them stupid and illogical too. Okay, so that's the furin cleavage site H5N1. What's the difference between pathogenicity and virulence? Pathogenicity is an all or none thing. You can cause, you're pathogenic or not. If you want to quantify disease, pathogenicity, you use virulence. Okay? So the virus is pathogenic or it's not pathogenic. And virulence measures the pathogenicity because it can fall in between, right? Okay. So viral virulence is the disease-producing power of a virus. We talk about virulent viruses that cause disease or attenuated viruses that do not, like attenuated vaccines, right? And we can quantitate virulence in many ways. If you're doing virulence studies in animals, you can measure virus titer, mean time to death, mean time to appearance of signs, not symptoms. Symptom is what a patient can report, and animals cannot report symptoms to you. So they're signs, things you can measure. Measurement of fever, weight loss, pathological lesions, say for polio in the central nervous system. Reduction in blood CD4 positive lymphocytes for HIV-1. So we count the number of of CD4 positive lymphocytes to get an idea of when we are approaching clinical AIDS. Many signs and symptoms of disease, in fact, are caused by the immune response. We'll talk a little bit about that in part 2. It's too much for today. Just remember that. Immune response is a double-edged sword. So here are 2 assays for measuring viral virulence. Pretty straightforward. On the left is whether or not the animals die. It's called a Kaplan-Meier plot. The number of survivors versus the day post-infection. And here we have infected mice with either type 1 or type 2 poliovirus. And you can see the mice infected with type 1 all survive. Whereas the mice infected with type 2 slowly die until there are none left on day 11. So by this assay, and we could say how much virus do you need to kill half of the survivors. That would be done by a titration study. But this is using death as a measurement of virulence. And here on the right, we use pathology to measure virulence. So we've infected, I don't know what we're infecting here now. I've forgotten. I don't know. It could be non-human primates. But anyway, with all these different viruses, and then we remove different parts of the brain, the cerebellum, the brainstem, and the spinal cord, and we make slices and look at them under a microscope and count lesions that are caused by the virus. So relative neurovirulence score is calculated based on the number of lesions. So more lesions, higher neurovirulence. So you can see Japanese encephalitis causes lesions throughout the central nervous system. A little less with yellow fever. West Nile is big in the brain. Langot is big in the brain and the spinal cord. And dengue is not neurotropic. It does not reproduce in the brain or spinal cord, so you don't see any lesions. So that's another way to measure virulence. And these are all flaviviruses, so that is doable. And that brings me to this. Viral virulence is a relative property. It's influenced by dose, route of infection, species, age, sex, susceptibility of the host. So you can't compare the virulence of different viruses because one of these things is going to be different. For similar viruses like types 1, 2, and 3 polioviruses, the assays must be the same. And for this reason, measuring virulence in humans is very difficult without challenge studies, which we can only do with a few viruses. And in fact, those viruses have to be avirulent or a very low virulence in order to be usable in a challenge study. So here's an example on how the route of inoculation influences virulence. This is lymphocytic choriomeningitis virus, a virus of rodents. It's often used in immunology labs to study the immune system of rodents to infection. So if we take 100,000 PFU of this virus and inject it intraperitoneally, you know what that is, right? Into the belly, into the peritoneal cavity, all the mice survive. Not a single one becomes ill. Look, lovely coat there. Looks great. If you put one PFU right in the brain, intracranially, you go through the brain, you go through the skull, which is you can do in young mice because it's pretty soft. You go right into the brain. One PFU, one plaque-forming unit, maybe one virus particle, one infectious virus particle. The mice die. All the mice die. So how you infect them really makes a difference to virus. So you got to specify everything. So here, for example, is what people were doing during COVID. Here's a paper out of Ontario. I'm sorry, our Canadian friends. This was published. Progressive increase in virulence of novel SARS-CoV-2 variants in Ontario, Canada. Nope. Wrong. Incorrect. This is an observational study where you measure hospitalization during circulation of different SARS-CoV-2 variants, okay? And they claimed that the virulence went up with each one, but they did not control for confounding variables like immune status, age, comorbidities, and who knows what else that would control virulence. They can't. It's an observational study. In fact, you can't conclude anything from this. So the title is wrong. You could say observations on the virulence in Ontario, but you can't say progressive increase. No. It's not possible to conclude that one variant is more virulent than another simply from observations of hospitalization. So you could use an animal model and look at it, but I don't know what that means for people. And you can't do it in people. So you see why it's hard to measure virulence in people. It's really, really hard in the real world. Now, in virology, we'd like to identify viral and host genes that determine virulence. Why? Maybe we can interfere. So what we do is we make mutant viruses. This is what we did when I was working. So I guess it's prehistoric era. Nobody does this anymore. So we make mutations in the viral genome and we look for viruses that cause reduced or no disease in a particular animal model. Okay? And so here's what you can do. You have a virus, a wild-type virus that grows very well in cell culture, makes nice plaques. You infect the brain of the mouse. It causes disease. It's neurovirulent. Then you make some viral mutants, and here you have a mutation that leads to a general defect in reproduction. You get small plaques, you get lower titers. You put this in mice, doesn't reproduce well. You don't get disease. Boring. Totally boring. We don't care about how growth affects pathogenesis. We know that would be affected by that. What we want is the third category where we have made a mutation in the genome. The virus grows well in cell culture, nice plaques. You put it in animals, it doesn't cause any disease. That is a gene that is specifically required for virulence, for causing disease. That's what we want to study. And so many people have over the years, and they've identified 4 classes of viral genes that regulate virulence. The first one, boring, genes that affect viral replication, genes encoding toxins. Not too many of those, but the rotavirus story that we talked about on TWIV, that's one example. Genes encoding modifiers of host defense mechanisms, right? Genes that antagonize host defenses. Those are definitely viral virulence genes. And since those genes, there's no immune defenses in cell culture, the virus grows well. And finally, gene products that enable virus to spread in the host. Now, mutations in these last two classes don't affect viral replication in cells in culture, and they're often called non-essential genes, which is a bad name. It's a dumb name because they have a function in the animal to antagonize the immune response or enable the virus to spread in the host, properties that are not present in cells in culture. Okay? I hope you're getting this. Now, these genes that we identify don't have to encode proteins. They can be non-coding sequences. And in my opinion, the most famous example are the mutations in the Sabin vaccines of strains of poliovirus with mutations in the 5' non-coding region, and they reduce neurovirulence. So here's the poliovirus genome. The 5' end is highly structured, and this particular stem loop 5, Sabin selected mutations in the type 1, 2, and 3 strains that reduce virulence. And I'll show you an experiment we did in my lab. We have two polioviruses that differ only at the one base in the 5' end, the type 3 base, which is a C in the virulent polio and a U in the vaccine. When you inject these viruses into mice right in the brain, the viruses with a U do not cause any paralysis. The viruses with a C, half the mice get paralyzed when you put 9,000 PFU in. And the viruses with U do not reproduce. This is virus reproduction in the brain with time after infection. Viruses with a U don't multiply. Viruses with a C do, and that explains why they paralyze. To this day, well, it took me many years. I worked on this for many years to try and figure out what these mutations were doing. I thought they were mutations specific to the central nervous system. Somehow they specifically regulated virus reproduction in the central nervous system. But no, there is no such thing. It's all about fitness. These mutations were selected by Albert Sabin to reduce virulence, to reduce paralysis in non-human primates. They do so by reducing the fitness of the virus. So you get less reproduction, less chance that the virus is going to get into the brain and spinal cord. As the virus reproduces in the gut, these mutations in the 3 vaccine strains revert, restoring fitness to the virus. And that's why those revertants are capable of causing polio, and they do. There's still most of the polio globally today is caused by vaccine-derived type 2 poliovirus, which has reverted here and at other places. Okay, we've identified viral gene products that modify host defenses. We have proteins that antagonize apoptosis and autophagy, intrinsic programs. These are proteins. These are all intrinsic defenses. We have homologs of chemokines and their receptors that interfere with chemokine function. We have complement binding proteins that prevent complement from working. We have modifiers of MHC1 and 2. Remember, these are the plasma membrane proteins that present antigen to T cells, for example, or to T cells. And these are often not required for growth in culture. So, you might call them non-essential, but that would be wrong because they're needed in animals. So here's an example of identification of a viral virulence gene using a mouse model of gamma herpes virus 68. This is a natural mouse herpes virus. And what they have, when you infect mice with this herpes virus, they die. So what was done here in this experiment is they deleted the M3 gene, which is a chemokine receptor homolog. It antagonizes chemokines. And then you do a percent survival versus dose of virus. So the wild-type virus in blue, the 50% survival is about 5 PFU here. And if you delete the M3 gene, the 50% survival goes way up over 100 PFU. So you've reduced the virulence of this virus by removing its ability to antagonize the immune system. That's an example of a viral virulence gene. The rotaviruses encode a toxic viral protein, NSP4, that interferes with calcium levels in the intestine, and that gives you the watery diarrhea that is characteristic of this disease. How about some host virulence determinants? So we've talked about viral so far. How about host? So herpes simplex, encephalitis. Many of us have latent. You get infected with your first bout of herpes viruses. They enter mucosal cells in your upper tract, and eventually they become latent in local ganglia, the collections of nerve cells outside of the central nervous system. And on reactivation, you get a cold sore. The virus goes back to the mucosal layer. But sometimes the virus goes the wrong way, and it goes to the brain and causes encephalitis, which is a potentially fatal infection. About one case out of 250,000 a year. And if untreated, 70% mortality. There are two peaks of incidence of herpes encephalitis between 6 months and 3 years. That represents the primary infection, where even then the virus can go the wrong way to the brain. And then over 50 years, this is reactivation from latency. You get reactivation in the ganglia. Instead of going to the mucosal cells, it goes to the brain. All right, so this is a nice number, so you can begin to ask what kinds of single nucleotide polymorphisms are involved in herpes simplex encephalitis. And so what was done was to take patients who had herpes simplex encephalitis and sequence their genomes, do genome-wide association studies looking for single nucleotide polymorphisms, comparing them to people who don't get herpes simplex encephalitis. And they identified mutations in several genes involved in innate responses to herpes viruses. In particular, toll-like receptor 3, which senses viral RNA, and a number of the other proteins involved in toll-like receptor 3 function. So what happens is these individuals have defective innate responses. The virus reproduces to high levels, and it enters the brain instead of going to the mucosal layer. So this is the beauty of genome-wide association studies. Another host gene that determines susceptibility is CCR5. CCR5 is the second receptor needed for HIV attachment. You need CD4 and CCR5, and it turns out that there's a mutation called delta 32 in CCR5 that protects against HIV infection, because viruses can't enter the cell. 4 to 16% of people of European descent have this deletion, and they don't get infected with HIV. Lucky them. You may remember the German patient years ago. He had both leukemia and AIDS. His physician had to give him a stem cell transplant to cure the leukemia. So what you do is you ablate the stem cells in the bone marrow. Those are the precursors for all your immune cells. And then you give a transplant from someone else with normal bone marrow, so it's going to make all normal cells, no more leukemia. The physician was smart. He read the literature, and he knew that people with CCR5, delta 32, would not get HIV/AIDS, so he found a donor for this German patient who was CCR5 delta 32, and the German patient was cured of AIDS. First such patient, and 4 others subsequently cured in the same way. But anyway, the point is that this host gene determines susceptibility to infection. Other determinants of virulence: age. Very old, very young and very old humans are the most susceptible to disease. Here's flu in the U.S. from 1911 to 1915. Death rate over age group: very young and very old, not very, but older, have excess mortality compared to the other age groups. So why is this? Well, the young don't have a very good immune response, maybe, and then when you're old, your immune system is senescing. You have less elastic alveoli, weaker respiratory muscles, diminished cough reflex, reduced rate of production of new immune cells because your bone marrow diminishes with age. That's why we think age is one factor. Now, the main comorbidity, the main factor for severe COVID is age. This is COVID case fatality ratio with increasing age, data from a couple of different countries. You can see with increasing age, the CFR goes up until 80 plus years, it's 15 to 20%. So age is a big factor for COVID mortality. Why is that? What underlies critical COVID-19? So COVID-19 is associated with about a 1% risk of death. It doubles every 5 years from childhood onwards. As I've shown you in that preceding graph, it goes up as you get older. And it's also 1.5 times greater in men than women. Now, in general, COVID is 40% asymptomatic, 40% of benign upper respiratory tract disease, about 20% of the patients get pneumonia, and 10% of them get severe pneumonia requiring hospitalization. One of the main reasons for severe COVID is about 4% of people over 70 make autoantibodies to their own interferons. So here we have a graph of age, and we're looking at the infection fatality rate on the right. And here we're looking at autoantibodies to type 1 interferon. And you can see the red are pretty stable until you get to about 60, then they go up, and that correlates with the increase in the infection fatality rate, or the case fatality rate. Inborn errors of immunity, those single nucleotide polymorphisms, also account for some of the severe COVID. And studies have shown that genes inherited from Neanderthals, plus 5 other regions we don't know where they came from, are mainly involved in interferon immunity, and those underlie critical COVID as well. So you can see we have a color code here. We have children, adults, and older individuals. We have inborn errors of immunity, which is pretty much constant among all ages. Then we have autoantibodies, which you can see go up significantly in older individuals. And then there's unknown risk, the gray, which also goes up with age, and we don't know what causes that. I think that's pretty cool. Other host determinants, men are slightly more susceptible to viral infections than females, but not always. Females generally have elevated humoral immunity, antibodies, compared with males. It seems to be phylogenetically conserved. It's in other animals as well, and probably is meant to ensure reproductive success. The females are better protected against diseases so that they can reproduce. And you can see in this graph that these are antibody responses to flu vaccination. And you can see that the adult females, they have better response than adult males. But if you look at older males and females, called aged here, you can see now the females and the males are more or less similar. And so the idea is that the elevated humoral immunity is needed to protect females until they give birth, they reproduce, and then after a certain age, it doesn't matter anymore, so the immune system deteriorates. And by the way, female antibody responses correlate with elevated estradiol. And pregnancy is also more lethal. Infections are more lethal for Hep A, B, E, influenza, and COVID-19 infections for reasons of immunosuppression. Malnutrition is another determinant of virulence, and that's why measles is 300 times more lethal in developing countries than Europe and North America. Cigarette smoking increases susceptibility to respiratory infections, air pollution, and stress. All of these increase susceptibility to infection. Okay, that's a part 1, mechanisms of pathogenesis. Next time is not going to be acute infection. It's going to be part 2 of mechanisms of pathogenesis. I really like pathogenesis, and I think it's understandable, right? You guys were chatting away while I was talking. I saw it. It's okay. All right, this is where I stopped with Noir. And here, MN says, please give Amy our best. So should she be listening? There you go. Examples where the drug didn't work in the animal models but worked in humans. Well, I think if it doesn't work in an animal model, that's it. Never going to proceed to a human trial, right? Because you can't ethically give people a drug that doesn't work. It could be that you could modify the drug to get it to work, but I don't know how that goes in companies. I was reading about Charles River Laboratories and their mouse models. Yeah, Charles River and Jackson Labs are 2 big mouse vendors. How are they being affected by the madness? Well, I haven't heard anything. They're companies, right? They could make mice. Now, they're going to be affected because labs aren't going to buy their mice anymore, right? The university labs that have their grants taken away, they're not going to buy mice, so their business is going to go down, and I hope they can stay in business because they have a huge resource of different mouse strains. Thousands and thousands of mouse strains are in existence. At Jackson Labs. Mice lie and monkeys exaggerate. That's right. You'll remember that. Do neurons not produce interferon? Yeah, they produce interferon. Then some immune cells get in. They get in and they produce interferon as well. So yes, brain cells do produce interferon. Mice like monkeys exaggerate and humans can only be observed. That's right. Brian wants to know if misfolded prion proteins are more rigid. Yes, they are more rigid, and so the native protein before it misfolds is completely digestible with a protease called proteinase K. And then when they misfold, it only cuts it in a couple of places because it's got a lot of beta sheet now, whereas before it didn't. That's right. One friggin' base causes such a difference. That always blew me away. One base. Is reversion like a reverse bottlenecking process? You know, John, it sounds very interesting what you are saying. So a bottleneck is when you pass a population, let's say viruses. You have a big bottle of viruses and then they pass through the neck and only a few can get through. And then what gets through is not as diverse as what's in the bottle, so we call that a genetic bottleneck. So when those viruses that pass through reproduce, they have a very different population structure than the ones in the bottle. So now a reversion is not really that. A reversion is simply when the mutation changes back to what it was. So remember, the type 3 poliovirus had a U at base 472. Sabin selected variants that had a C, natural occurring variants. And then when those go in your intestine, they revert to U. They revert to C within a couple of days. Yeah. Tryon wants to know, was SARS-1 more fit to affect lungs compared to SARS-CoV-2? Now that's an interesting question. So I think the main difference between SARS-1 and 2 is that there was during the asymptomatic period, there's no viral shedding for SARS-1. So people who were still well, who were infected, were walking around, but they weren't transmitting virus. And then when they started to shed virus, they were sick. They were really sick. And they had a higher rate of mortality in SARS-1 compared to SARS-CoV-2. Now, SARS-CoV-2, there is shedding and transmission during the asymptomatic phase. And so that is why the virus so effectively spreads. You have people walking around who don't know they're infected, and they're shedding and transmitting. Visto wants to know, would a polio mutation fit in the gut but not in the CNS be possible? So that's what we always thought, right? That's what we always thought was the case, that there was a specific... So our idea was that U could replicate in the gut but not in the CNS. And, C could replicate in both places. That's why C would cause paralysis. But it's not the case. I don't know if you could make one that would be like that. Maybe you could. But, NOPV2, the modified OPV2 that was made by molecular virologists, they made changes in the 5' end that prevent reversion. Those viruses don't revert in people. What they do, the whole 5' end is recombined out and replaced with a different enterovirus, and it makes it more fit. So there's no specific sequence because the other enterovirus is totally different. It's just a matter of restoring fitness. Yeah. Kang wants to know, is CCR5 an example of lack of permissiveness? No. CCR5 deletion, delta 32, is lack of susceptibility, right? Remember, susceptibility has to do with presence or absence of receptors. So Mark gives us a little lesson about prions. Beta-pleated sheets act as a catalyst to misfold other proteins, and this process amplifies prions. As the protein stack becomes more difficult to break down, cooking infected meat does not clear prions. That's why people got it from hamburgers in the UK in the 80s from meat from cows that had mad cow disease. Brian wants to know if virus-infected cells increase metabolism and decrease pH can be explained the use of urotropine decades ago to treat viral encephalitis. I don't know what urotropine is. Let's look it up. It's used as a urinary antiseptic. It's used industrially as in the production of resins and as solid fuel. So I don't know why you'd use it for encephalitis. I'm sorry, Tryon. I don't know. COVID variants on perplexed that males are more vulnerable to the virus. They're more vulnerable to lethality, death, right? But females are more vulnerable to long COVID. Yeah. I don't know why. Nobody knows. Patricia wants to know, can you state why the following is or is not true? We need a booster better matched to the version of the coronavirus currently circulating. Okay, I did a mini lecture on this a couple months ago, but I'll do a mini digest of that. So let's say we have a new variant circulating and the CDC approves a vaccine that matches it. And a bunch of people go get vaccinated. They've already been vaccinated. And so if the variant is sufficiently different from your vaccine, you will make a new immune response to the variant. If it's very close to what you were vaccinated with initially, then you're mainly going to make an immune response to that original vaccine. But let's assume that it's different enough. Now you get an immune response that's going to protect you for a couple of months at most at severe disease caused by this new variant, right? But in a couple of months, your antibody levels and T cell levels are going to go down, and you're going to get more moderate disease than you would before. And if you're older, over 70-ish, say, you could get hospitalized. Even though you had this matched vaccine, the fact that it viruses. So the problem is you need a memory response, and that takes a couple of days. And for some people with comorbidities, that'll throw them in the hospital. That's a couple of days of virus multiplication. So basically, you're getting 3 months of some kind of protection. That's why the CDC is making these recommendations. So you need to decide what you want to do. I have not had an updated COVID vaccine in a while. I got the original 3, and I feel I had COVID once and I took Paxlovid, and it took care of it. So I feel if I get it again, I'm going to take Paxlovid. And other people can decide what they want to do. But, for older people, maybe 3 months of protection is good. Or if you want to go on a trip, maybe that's good protection to get vaccinated a couple of weeks before the trip. Okay? Can virus infection induce prion disease? I'm not aware of that, no. But you know what? Anything is possible. Who knows? I like this Vista. I understand almost all of that. Not so many acronyms in parthenogenesis. I thought he was making a joke, which would have been funny, but it was a autocorrupt. From bi-mice to bi-mice. That's right. Kang wants to know, is the higher antibody levels in women needed to give passive immunity? That's a good idea, Kang. I mean, I don't know this field all that well, but Savra Klein was on TWIV a while ago talking about sex-based differences in immune responses. And that makes a lot of sense. I like that. I don't know the answer, but I like it a lot. A and V have a lot of mouse husbandry knowledge. Yeah, we did it. So my career, I did a lot of mouse work. And then when Amy came in her last couple of years to Columbia, we worked together because I had a lot of mouse experience and she wanted to do mouse experiments. So every week we'd go to the mouse house and inject mice or bleed them or whatever. Yeah. Interference seems to be a bit like steroids in reverse. Dangerous in the first week, but helpful in the second. Well, actually, you need the right amount of interferons in the first week, right? And if you have, if you have too much, well, if you don't have enough, the virus is going to multiply like crazy, and then you'll get more interferon, which then in the second week will give you an inflammatory problem. Okay, so the first few days, the interferon level is critical. You need to have the Goldilocks amount. Yeah. Mark is asking whether the virulence of SARS is partially due to misregulation of ACE. I'm not aware of that. I mean, it's an interesting idea, but I'm not aware of any studies that support it. No. Will says 3 months of protection due to a booster. My anecdote is that I got my second infection 4 weeks after my booster. It can vary, yeah. So how was your what was the severity of your infection? Was it, would you say mild, moderate, or severe? I presume you didn't have to go to the hospital, so it's either mild or moderate. I'm curious. But in some people, everybody has a different kinetic of antibody response. And so I'm not surprised that, 4 weeks you got infected. Anybody have any questions? It's 9:40, but we can stop if you're done. Let me know. What does furin do? Furin is a protease that cleaves proteins that have a furin cleavage site. I mean, it has functions in the cell. It regulates the processing of proteins. Sometimes you don't need a protein any longer. It has to be cleaved by a furin to make it functional. So lots of functions in the host cell like that. And of course, the viruses are just tapping into it. So Will says the second infection was pretty mild. So maybe maybe that was your memory response to, or maybe you still had good levels of antibodies, and that made it mild. But that's good, right? Maybe if you hadn't had the vaccine, it wouldn't have been mild. So Kang wants to know, how did Pasteur develop a rabies vaccine without knowing how rabies was caused by a virus? Well, he knew it was an infectious disease, right? Because Koch had already shown that microbes could cause diseases. So Pasteur thought it was so back then, anything that caused an infectious disease was called a virus. So Pasteur assumed it was a virus, which basically meant a bacterium. And he had the idea of vaccination, right? Because what's his name? The guy who developed vaccines, who made the first smallpox vaccine. I can't remember his name. But Pasteur was aware of those results, and so he said, I can make a vaccine for rabies. And so he took infected tissues and activated them in various ways in that way. And, basically what you have is you have an animal model for rabies, and then you put your vaccine in and you see if it works. Edward Jenner. Thank you, John. If I don't use a word very much, I forget it. I'm glad you liked the mini lecture. Thank you very much. It is, I try and keep it a little more simple here, right? For this crowd. I know you're a scientist, Zhu, but I try to keep it simple, and that helps everyone, including myself. Tom writes, certainly one expects recombination in an enterovirus, but what amazes me is recombination among different enteros. Yeah, so the poliovirus is when you take the oral poliovirus vaccine, they go into your gut and reproduce, and then you have a lot of different enteroviruses, but it's only the type C enteroviruses, a certain grouping of enteroviruses that will recombine with poliovirus, but they're not polioviruses, right? They're just other kinds of enteroviruses, and yes, they recombine and replace that whole engineered 5' N as well as the polymerase, and the virus has regained fitness. It's amazing. Yeah. Yep. Why is it that babies don't learn immunity from antigens? Babies' immune system isn't developed until about 6 months, so it can't recognize antigens. And so what the mother does is provide antibodies that, at birth you get all you're going to get from your mom, obviously, although you can get some from breastfeeding, secretory IgA. And then the mom's antibodies slowly decline, and then your system kicks in, right? And that's how it works. Noir wants to know the difference between gram-negative and gram-positive bacteria. So they're bacteria that differ in their cell wall structure, essentially. So the gram-positive bacteria have a thick, it's called peptidoglycan, right? It's protein and sugar. And and gram-negatives have a thin peptidoglycan between an inner and outer membrane. And so gram-positive, so you use a stain that the gram-positive, that thick peptidoglycan will stain purple. That's a gram-positive. And then the gram-negatives will not pick that up. They'll just appear pink because there's a counter stain that makes them pink so that you can see them. So that's the functional difference, right? And so they're, like the E. coli's are gram-negative and the streptococci are gram-positive. So different classes of pathogens. So when you have an infection, one of the first things you can do is a gram stain to narrow it down to what it might be. Yes, Jenner, Edward, not Caitlin. Yes, Edward Jenner. So apparently Costella's child had a second place in a science fair. That's great. The radishes died, so we did a salt sugar dissolving thing. I think great resourcefulness, right? Cool. Chillin Lillian says bottlenecking is a really fascinating concept. Does it seem to imply that having the best escape sort of mechanism precludes fitness? No, in viruses, all it means is that a diverse population of viruses is needed for fitness. And by diverse, I mean where every virus genome is in a virus stock is a different sequence. And when you push a stock of viruses through a bottleneck, you're restricting the diversity of the population, and diversity is needed for viruses to do well. So what is a bottleneck, for example? Well, when a mosquito bites you, it's only giving you a few thousand viruses compared to what's in the mosquito. So that's a bottleneck. When you get a respiratory droplet infection, not many viruses in a droplet, so that's a bottleneck. Those are all cases where, bottlenecking happens. File 1271 wants to know if a COVID booster 6 months is beneficial for someone in their 40s. I think the CDC says anyone can get it. Is it beneficial? Well, there is an impact on long COVID of vaccination. It's a certain percentage of decrease of long COVID by vaccination. And so if that scares you, then it's beneficial. But in your 40s, you are healthy, and theoretically, you shouldn't need a booster. So speaking scientifically, not giving any medical advice, right? Questella, thank you for your support of science communication. Thank you so much. Appreciate it. Rainey says, my hands are still stained from gram stains 40 years ago. Chillin says, by escape mechanism, I mean some aspect of the virion which aids in the particle's ability to be pushed out of a densely packed environment. No, that's not what it is at all. It's just a matter of restricting population to a small number. For example, if you have a plaque and you pick it, you only have a few thousand viruses. And if you use that for something, you have a restricted population, and it may not behave as you think. But it has nothing to do with physical being pushed out, no. How many words and acronyms do you think there are in virology? I don't think there are 10,000. I think there are a few thousand, probably. And new ones are arising every day. So, yeah, that's an interesting question. Weaselbox, hello. Nice to see you. Mini lectures are great. Cut them up and serve to TikTok. Yeah. It's that would be good. How are HeLa cells in labs used? Well, as you know, they're derived from the cervical epithelium of Henrietta lacks, and they're transformed, which means they live forever. So they have the wrong number of chromosomes. They're abnormal. They're basically cancer cells. So you have to be careful what you use them for. You can't use them to study normal cell processes because, well, you can certain ones, but mostly they're used in virology to grow virus stocks and to titrate them. We used HeLa cells for many years. We did biochemical experiments. We would study the translation of the genome in HeLa cells. That's a basic process which it almost doesn't matter what cell you do it in. And then at some point in my lab, we shifted to studying the innate immune response, the interferon response. And I left HeLa cells in, and the Durant reviewer said, "What are you, crazy? You can't study interferon in HeLa cells." And I said, "Yeah, you're right." It has nothing to do with where the virus reproduces, right? So when you want to do pathogenesis, you have to use cells that reflect where the virus is, not HeLa cells. Jill, you have COVID and it's awful. I'm sorry. You'll get better. You're young. Bottleneck, Nevitz says Bottleneck is also a famous region on upper reaches of the Himalayan mountain K2, where many tragedies have occurred. Yeah, that's an idea. It's a thing it's hard to get past. So if you have 1,000 people, a gate is a bottleneck. It will only let a few through. Jason wants to know if immunity develops in other mammals similar to humans. Yes. In fact, if you go far enough down or left on the tree of life, lampreys, right? Those odd fish that they have a mouthful of sharp teeth and they attach to sharks and get a ride with them. They have single chain antibodies, and they work pretty much like our antibodies do. Yeah, so mammals are all pretty similar. It's then when you get into like insects, they have mostly nucleic acid based immunity, not protein based like we have. Anyway, yes, Jill, hope you get better soon. Okay? It's too bad you didn't take Paxlovid. This is funny. It would take a turn if viruses went from bottlenecking to rubbernecking. That's great. I love that. And Questella still is a novid, as far as you know. Good for you. Congratulations. Hello, Sandra. Yeah, you're late, but you can always go back and read it. Nevitz, do you recommend Reut's Essential Immunology? So I have old edition. I found it not so easy to read. What would I recommend instead? I don't know, but maybe the newer ones are better. Janeway is another good one, right? That's recommended. Weaselbox says, I share some of the coolest information. In my class, I always used to say, and I don't do it anymore, I don't know why, that every lecture you're going to have something you could go to your roommate and wow them about. And sometimes I would say, this is the wow moment, get ready. Like, all the phages in the ocean could go 200 light years into space. That's pretty wow. Okay, folks. Time to wrap it up. It's fun again, as usual. Hope you enjoyed it. I want to thank our moderators tonight. We had Tom Steinberg. We had, I know Steph was here. I'm just scrolling up the list. I know Les was here. I know Vanity Nutrition was here. Anyone else? I don't think Frank was here. I don't think Barb Mac UK was here. She was on Tweevil. Thank you, moderators, for being here tonight. I really appreciate it. We'll be back next. And thanks, everyone, for coming. We had 155 people, 137 likes. Not bad. We'll be back next week. Let me tell you the date so you can mark your calendars. So today is the 23rd, right? Oh, next week, it's the 30th. We're still in April. April 30th, 8 p.m. Eastern. We'll do another bout of this. Thanks for coming, folks. I really like your presence. Obviously, I couldn't do it without you. I'd just be talking to myself. Well, that's a thing, I guess, right? Anyway, have a great week. And until next week, please be safe. Thank you and good night. Bye-bye.